Chapter 2, going into the cell. Remember, cells are going to come together and form tissues, but cells are like the first thing that we can really study. We can see it under a microscope. Okay? So they are the building block of us. We have trillions of cells throughout our body, okay? Everything is made up of cells, all living things. Now, plants and animals living. Okay, so cells are produced by, they divide. Pre-existing cells divide and they make more cells, daughter cells. We'll go into some of that stuff, the mitosis and meiosis, in just a little bit. Okay. Our sex cells, those are the ones that are our germ cells. Okay, so in males it's sperms, and females it's the oocyte. The only thing you need to understand about that is when they divide, they go through a second set of division, so they only have half the chromosomes. So because they have half the chromosomes of the parents, when they come together, they have a complete set of chromosomes. Half from mom, half from dad. There we go. So we're all going to have the same amount of chromosomes. Somatic cells, they're all the cells that aren't sex cells. Pretty simple. And as you can see right here, here's an oocyte, and all the sperm right there is trying to get in there and doing their best to try to make it so we're going to breed. Parts of the cell. So we have our cytoplasm. The cytoplasm means inside the cell. The cytosol is the liquid in the cell. Organelles are going to be the organs, I don't want to say they're organs, but they are kind of like the organs. They are the substances within the cell. Okay? And then we have our cell membrane, which actually is the wall that is going to say, hey, this is a home. So a cell membrane is referred to as a plasma membrane. Then we have our cytoplasm, which is going to be all this stuff in here. Kind of like having this drawing feature. So the cytoplasm has our cytosol, which is all the liquid, and it's kind of the organelles, which are inside of the cytoplasm. They're kind of like the organs. These are all the parts of a cell. We'll go into more detail on these things as we go along with this class. This actual one right here. Okay, the main things about a plasma membrane are it's going to provide the physical barrier. So it's going to separate out the inside of the cell from the outside of the cell. It's going to regulate. So the plasma membrane, it is a phospholipid bilayer, which means it is made up of a phosphate and a fat. So the phosphate, it loves water, fat hates water. We've all seen what happens when you put oil with water, it separates out because oil doesn't want to mix with the water. But the phosphate loves the water. So the phosphate is going to face towards the water and the lipid is going to face away from it. Now by having those guys right there, it makes it so it's two layers. One layer facing outside of the cell, one layer facing inside the cell with the lipid legs kicking at each other. So that's why there's a phospholipid bilayer, two layers. So that makes the wall, and in that is called a semi-permeable. Semi means sort of. So it allows things in and out, but it has it's like the bouncer at the gate. Okay, it decides what gets in and what gets out. So it's responsive to extracellular fluid, and it's a way for cells to communicate from cell to cell. We'll go into more detail on that. Okay, components of the membrane, phosphates, proteins, glycolipids, and sterols. So glyco means sugar, lipid means fat. So sugary fat, kind of like cake. So these things that stick out, so right in here, what we're looking at here is going to be this right here is our phospholipid bilayer, right in here. These right here are going to be some of our channels. Now some of these channels are gated, so as you see right here, this right here is just free. Things can go in and out. This guy right here, these guys right here, they sort of touch. Looks like an H. This gate will open and let things in or let things out how it wants to. Now these guys out here, these are like always patrolling, and a lot of times what they are, they're going to be receptor sites. So something can bind on this and trigger a reaction, or I can bind on this, it can make it sort of want to take it in. So these are your receptor sites. Okay, phospholipids, we already talked about that. Here's that nice phosphate head. Here's the lipid tail. Loves water, hates water. This is facing towards water. Remember, we have, that's all water that way. This is all water this way. So these guys love it, and they're going all over it. So what we basically have right here is These guys right here are, they're happy because they're looking over at the water. These guys right here, they're sitting there dancing around with each other, so they're kicking their legs at each other. All right, that's all there is to it. This is a cholesterol right here, and if you take a look, it's kind of in the shape of bacon, so that's what you can always understand. Cholesterol is a main component of our cell wall. Okay, peripheral proteins, we've already talked about those. Those are on the outside. So up in this area, these are going to be more of your peripheral proteins. Our integral proteins, they're the ones that are in the membrane and they make the channels. So this right here will be an integral protein. Okay, that's all you need to be aware of. Oh yeah, cytoplasm. And this is, so we have our intracellular, so this is going to be our intracellular, and this is going to be our extracellular, so outside. As you can see right here, peripheral protein is on the outside, not the outside of the cell, but on the wall. So it's on the outer portion of the cytoplasm. I'm going to go into too much detail. A sterol is like a cholesterol, it's a fat. Steroids are made up of sterols. Steroids, they actually go right through the cell wall because they are fat-based, cholesterol-based. Permeability is how well things can move in and out. So we have passive and active, and we'll go into those things, all right? Impermeable means it can't go through. Freely permeable means it just goes in and out without any problems. Oxygen just goes in and out without any problems. Selectively permeable means the cell membrane, those channels are going to decide what it wants to have in and what should leave. Okay, so selectively permeable. This right here, I want you to think about this, is the guy at the velvet rope at the nightclub. He decides who gets in and who doesn't. Now, there's different ways to get inside. We can have diffusion, which is just real simple, no energy. Osmosis, which is the movement of water, or facilitative, where you actually need a protein to get in. Active means that the cell is going to actively try to get something inside, or try to get it out. Active means energy was spent, okay? So on active right here, just understand there was energy, which was ATP. ATP is energy. So active transport uses ATP. Endocytosis is bringing something inside. When you get an endoscopy, they bend you over with the camera and they go inside you. Endocytosis is bringing something in. Exocytosis is something exiting. That's simple. So here you go right here. Simple diffusion. We have a high concentration outside, low concentration inside. Easily passes through. Facilitated diffusion means that we have to have a carrier protein. Something's out here that we want to get in, but we don't want it by itself. Okay? So there's a protein that attaches to it and helps to bring it in. So it's brought in here. Okay. Let me see. Moving on. Okay, still no energy was used. And active transport means we actively had to have something come in. So we used energy for it. Okay. All right, best way to explain this is a nightclub. So you have a nightclub in Las Vegas. We have a casino. In that nightclub, when it first opens, there is very few people inside the nightclub, but there's a lot of people in the casino. So they don't charge a cover charge. What they basically do is they just have their doors open and people are free to come from the casino inside. High concentration of people outside, low inside. Diffusion means that it's just going to actively flow in. So the people diffuse into the casino. When the casino gets kind of full, people can say it's getting crowded and they can easily leave. Okay, they can diffuse in or diffuse out. So when the casino gets too crowded, when the club gets too crowded, people are all crammed together, but there's more space outside the casino. Some people will decide to leave because they don't like to be that crowded. So that's diffusion, high concentration to low. Empty club, busy casino, everything goes inside. Now, facilitated diffusion, what happens now is the club's getting more popular. So now the club is going to say, hey, you can't come in unless you have something we want. Now Vegas is specific. What Vegas does is they like to have girls that are very attractive and guys that want to just spend money. So what they will do is they will actually, when a guy shows up to the club, if he has a girl with him, they're like, we want his money. So she's the carrier protein, or vice versa. Hey, I'm not going to say anything. You have a sugar mama. So basically what happens is you have a rich individual and they have somebody with them that they're going to spend money on. So that is their carrier protein. So now the club says, hey, come on in. We know you're going to spend money. We want your money. Okay? So what happened now was the pretty girl facilitated the guy being able to get inside. They don't always stay together when they get inside. So you don't understand that. But the club's going to get their money. Now, when it comes to, these are both passive, no energy. When it comes to active transport, that's when the Vegas club is starting to get a little bit dead. Okay? It's seen its prime. Now it needs to try to get people, and it needs to entice them to get in. So now what it does is it hires people. So now it hires some girls to stand outside of the club and try to hand out some drink coupons for these guys to try to get them to come in and spend their money. What's going on now is the club is spending money, which is like ATP. So now it is using energy to try to actively bring people inside to get them to spend their money. So that's going to be your active transport. Okay. This is the book way of explaining it. Now, endocytosis is what happens now is we have a substance that the cell wants. That substance comes in, and what it does is it binds with the protein outside. That receptor site says, hey, we want you in here. So now it attaches these guys. What it does is it tricks them, and it brings them in. And as it comes down, the membrane wraps around them and it forms a vesicle. This vesicle now, or a vehicle, travels over to the Golgi apparatus where the product inside gets dumped in. I think it's modified, does whatever it wants, right? Now, on exocytosis, what happens now is it is going to be processed at the Golgi apparatus, and it's going to float through the cytoplasm until it hits the cell wall. It's going to spread out. It's going to kick it out of the cell. All right? Easiest way to understand this is, okay, you go down to Mexico. You go down to Mexico. I'm not going to say anything bad about Mexico. I love Mexico. But let's say you go down there, you're in a bad area. You got the cartels there. So the cartels, they see you walking along on the streets, and they see you have, a lot of money. Okay, you're flashing around stuff. You're wearing, Gucci's, Armani's, all that stuff. What happens now is you come over to an area, the cartel kind of tricks you. So it gets over to a room. You open up that room, and as you're opening it up, some guys grab you. Next thing you know, you're wrapped up. They throw you into that white van. I don't know why they always kidnap you in a white van. That's a vehicle. They drive you now to the safe house. While you're at the safe house, that's going to be the Golgi apparatus. So what happened now is you were just abducted. So now you have been endocytosed, brought in. They're going to now ransom you. In order to ransom you to prove they're serious, they cut off your pinky. So now they have modified you. They get what they want, and now what happens is they put you back into that van, that vesicle. They take you back over to where they found you. They open it up, and they throw you out on the street. Because now you're like trash. Okay? But you've been modified. You've been changed. So that is exocytosis. They're getting rid of you. Let's see. Got the same thing here. Osmosis, just want you to be aware of, is that's just the movement of water. Okay? Everything wants to have the same concentration. Water is going to be, it flows wherever there is high concentration of substances. So if there's a lot of salt, water flows towards salt. Okay? Just understand it like that. The main thing I want you to understand is how water controls salt, how salt controls water. Microvilli, don't worry about that. We'll talk about that stuff. Cytosol, we just talked about. Kind of be aware of this a little bit. There's more potassium ions inside of a cell than there are outside. What that does is it helps to just keep the charges positive, the way that they want. Okay. Worry about the membranous stuff. Okay, let me just fix this up for you really quick because you need to know what this is. This is a really quick cheat sheet that I put together for you. I don't know why it all of a sudden decided to go on 3. Yeah, that's a little bit big there. Can't go wrong with 12. Okay, so I'd be able to read this. Just telling you what the parts of the cell are and what they do. Let's not go into too much detail on that stuff. I think it's really good for you to know, but just study up on this really fast. Okay, cytoskeleton is just kind of like the framework of the cell, which gives it its shape. See, centrioles, the only thing you have to worry about that has a flagella is basically going to be sperm. Centrioles, we'll talk about those as we go into division. Okay, ribosomes I want you to be aware because what ribosomes do are they make the proteins. So they get the code to make a protein, and that protein starts to get built in the ribosomes, and then it gets folded and put together into an actual functional protein. That's all ribosomes do. Ribosomes are what the coronavirus are, a single-stranded RNA. It takes over the ribosome and it starts to make the ribosome code the proteins that are making the RNA, because RNA and DNA, they're just nothing more than protein bases, okay? So they are the bases that make protein, okay? Go into detail on that in a little bit. See, mitochondria, just know that's the power plant. The nucleus is where the DNA of the cell is. That's the headquarters. Okay, Golgi apparatus like the UPS. That's the packing and shipping center. Lysosomes, what happens there is if there's a problem with the cell, lysosomes will actually go and they'll release enzymes that will break things down. Sometimes it will actually lyse the cell. So it destroys the cell, it was lysed. We named Lysol after that. So Lysol disinfectant spray because it kills stuff. So lysosomes kill. See all about that. Flow. Okay. I want you to understand that cells are going to be packed in kind of close. Because they're packed in close, they need to have a way to get nutrients through to the next cell over and waste away. So what we have here are these little gap junctions. So these gap junctions here, as you can see, we have a cell right here. And we have a little space between the cells. Over here will be another gap junction. So it's right in this section. You can actually see the little space in between the cells. You see these little spaces here. And they are anchored together. Okay, so we actually have these junctions that anchor those together. What we want to do is make it so it's not, things aren't going to be too floaty, but we can actually get the nutrients from one cell to the next cell to the next cell. So like this cell right here, it can get some waste products, you can push through here and it can go out. This one can absorb nutrients from this guy. We have a lot of blood vessels, and blood is what carries all the nutrients and waste. So the cells will have to get nutrients from the blood vessels and waste to the blood vessels. But the cells, remember, they can't move around. So they have to have a way to get things through. So here's our gap junctions. Okay. Let me see. They're referred to as like desmosomes and stuff like that. But desmosome just means that things can just go through, like they destroyed a little hole. That's all I really care to understand with that. Let's see. Okay, this is going to be what happens in a cell life cycle. So this area right here is going to be when the cell is just kind of hanging out and doing what the cell is supposed to do. So this is kind of where it's chilling and relaxing. Okay, this is sort of its job. We go here to the G1 function. Okay, this is where the cell starts to duplicate some organelles. It goes into a higher alert. What basically it's saying is, hey, we're going to grow, we're going to divide. So this right here is going to be our preparation phase. Okay. Now over here we start to make our DNA. So right here you're going to start to see the DNA replication. It gets bigger. Over here we have more and more proteins that are being made. So the cell is getting more compact. It's getting more crowded inside the cytosol. So this right here is all preparations for the big climactic split. Okay. See, the best way to understand this is where a couple is going through all their fighting and everything, where things just aren't going on and they're about to start giving up. Right in here, the DNA, this is where they're deciding who's going to get the kids. All right. Now over here, this is where we start to have to decide who's going to get all the stuff. Okay. Now realize, this is a nice way to look at it because Right here, we spend more time trying to save it. Over here, we spend more time trying to figure out who gets the kids. Over here, we spend a little less time trying to figure out who's going to get the car. Now over here is when we go into those proceedings with the lawyers, where they have to start going and paying and doing everything and separating everything out. Okay, so I've talked about all this stuff. What about this? Here's where I want you to be. So these right here are the steps to actually cell division. So this is your mitosis. It's called cytokinase because it's, don't worry about it. Cytokinase is just the end portion of making a new cell. So best way to understand this is real simple. We have our prophase. So prophase, we've gone through our interphase where the cell is just doing what it's supposed to do. We've already decided, so right here the cell is just living. Right here the cell is getting prepared because it's fighting and everything and knows it's going to split up. Right here it's deciding what to do with the DNA, deciding what to do with the kids. Now it's preparing to split up its stuff. Now we're going through the split. So prophase, that's your first phase. Now in prophase, you're actually going to see, so our hands right here, our fingers are going to be our DNA. These are our chromosomes. We have our chromosomes, they've duplicated. They're all sitting over here. In metaphase, they've lined up in the middle, and our elbows right here, these are going to be our centrioles. Your centrioles are like cowboys. They actually have lassos that come over and they attach onto the DNA. In our anaphase, we have now had the centrioles. They have now pulled apart our chromosomes. So now they have pulled apart the chromosomes. Now in telophase, the cell walls have now split up, and now what we have are two new cells. So prophase, we've duplicated our DNA. Metaphase, I line up in the middle, and our centrioles went to the side. Anaphase, they pulled apart. Telophase, two new cells. There you go. And that's what it is without the sign language. Right? Hope this wasn't too fast. Kind of an interesting.